All right, that's enough from me. We've got a couple of great keynote speakers, Dr. Aaron Lun and Laurie Shepherd. Laurie will be presenting tomorrow, but this afternoon, we've got Aaron. So Aaron will be familiar to many people here. He did his PhD here in Melbourne at the WEHI with Professor Gordon Smythe, before doing a postdoc in the UK with John Marioni. Aaron is a prolific bioconductor developer, answerer of questions, and general advice on all things RNA in particular. Aaron's currently working at Genentech in the US, and he's come all the way from San Francisco for today's talk. So big thank you to Aaron, and a big thank you to Genentech for implicitly supporting that. And over to you. Please. I still have that jump
All right. Oh, so fancy. You want to hear me? Okay. I think it's the second one. Ah, yes, this looks familiar. Okay, how do I? Okay, let's see. Oh. All right. So, hello everybody. Thanks to the organizers for inviting me here today. It's nice to be back in Melbourne, even though the weather's sort of a bit dodgy, but that's fine. Yeah, so today I'm going to be talking about some of the work that Jay and I have been doing in taking a single cell pipeline written in R and getting it to work inside the browser. Now, before everyone starts groaning, this will not be another single cell talk. I promise you there will be a minimal discussion of statistical methods or biology. So the statistical methods used here aren't novel, and they're covered in the Oscar book that was published last year. So the link's over there. And thanks to Pete and others for taking over its maintenance, actually, because that's a lot of work. As for the biology, I've forgotten most of my molecular biology courses, so there's really nothing to fear from me there. So the contents of this talk will be kind of like this cake. So most of it will be programming. There will be a thin icing of statistics. And if you're lucky, you might hear me mention 1 or 2 gene names, and that's the biology on the strawberry here. All right, so for those of you who have done single cell analyses before, you know that the tutorials make it look so easy, right? It's a very straightforward linear flow of steps, starting from loading the count matrix, quality control to remove damaged cells or poorly sequenced cells, normalization to remove cell-specific biases, selection of interesting features, usually based on highly variable genes. Then you've got some principal components analysis to denoise and compact the data further, and then there's some clustering on the PCs to get some interesting subpopulations, as well as some more artistic forms of dimensionality reduction. Is this black box going to be there for the entire thing? Okay, hold on. Let's see if we can move this. Yeah. Okay. We've all seen enough of the Roche logo. That's fine. Okay. No, this is recorded. Okay. Yeah. Anyway. So yes. At the end of all this, we get results that look a bit like. Okay. Yeah, okay, there we go. We get results that look a bit like this, right? You have a pretty picture with some clusters of interest, and then you can go ahead and assign each cluster to a putative cell type using either manual inspection of marker genes that you know are expressed in each cell type, or using automated classification methods if you have some reference data sets. Of course, in practice, it's not so straightforward. And that's because each analysis step has several degrees of freedom in its parameterization that can determine how you can proceed with it. So I've listed a couple of the choices that we might have to make at each analysis step here. For example, the resolution of the clustering will determine the granularity of the putative cell types that you end up with at the end of the analysis. So in general, it's not always easy to know whether a particular choice of parameter is sensible ahead of time. And in fact, there might be multiple sensible choices of parameters if different parameterizations give you different perspectives on the same data set when as you explore the data. Yeah, what I want to get to, so basically what this means is that many people will, repeat the analysis multiple times across several iterations in order to settle on a result that best addresses their scientific question. So basically, this means that these single cell analyses are often semi-automated. You can mostly run them in an automatic fashion, but there are times where you have to have manual user intervention to fine-tune some parameters depending on what your question is. And as we can see from the ever popular t-SNE and UMAP plots, there's a strong visual component to the interpretation of the results. So what software platform or environment or framework allows for this interactive computation as well as visualization? Well, there's R, of course, as well as Python and Julia and all of that. But there's a much more, in my mind, there's a much more obvious choice, and that is to create a web application. So web applications provide a very nice way of creating a user-friendly analysis environment. So web apps have strong support for visualization and intuitive interactivity that's been honed over several decades of user and developer feedback and experience. All the user needs to interact with a web application is a browser, which is basically installed on every consumer device these days. And in fact, the users themselves have effectively been pre-trained to know how to navigate a web page and click on the buttons and drop downs and all that stuff. People know how to do that already. So there's no need, if a user wants to do an analysis and you have a web application, there's no need to teach them programming principles like we would in R and Bioconductor, because all they need to do is point and click, and they know how to do that already. So the use of a web app for bioinformatics is not really a novel concept here, either for bioinformatics in general or for single cell in particular. So for example, the Galaxy provides a web interface for many bioinformatics workflows, and for everything else, there's like millions of shiny apps, of which I am guilty of authoring at least a couple of them. However, the thing that almost all of these have in common is that they all use, or almost all of them use, a backend server to do the calculations. So in a backend setup, we have the client, which is the user on their laptop or desktop, opening the browser and visiting a web page. And then when they want to do an analysis, the browser sends a request to a server where all the analysis software is stored, be it in R or Python or C or some sort of command line tool or whatever. This server or backend then does all the calculations, does the heavy lifting, and then once the result is obtained, it sends the result back to the client, back to the browser, so that the user can visualize it and interact with it and so on. And this cycle then repeats upon the next round of user interactivity. So this is a pretty popular model. This is a pretty popular architecture because it abstracts away the details of how is software installed, what resources does the server need. The user doesn't have to care about any of that because all they do is open a web page and the server handles the rest. However, there are some difficulties with this approach, and the most obvious of these is the fact that you need to spend money to set up a server and to keep it running. And if your app becomes even moderately popular, you need to spend even more money to make sure that the server has enough resources in order to handle the increased usage. So be that a physical server or an EC2 instance, you still need to spend some money. The need for network communication also means that you need to transfer data to the server, and then you need to transfer results back from the server. And that can introduce some latency, which makes the app feel sluggish and unresponsive, especially if you've got a lot of data and results moving back and forth. It also makes the app quite vulnerable to network outages or bandwidth throttling or other sort of arcane network details that I don't really know too much about. And finally, I put a picture of my favorite James Bond here to remind me to talk about spying. So if you have some data and you want to analyze it on an application that uses this backend setup, you need to transfer your data to the server so that the server can do calculations on it. And as soon as you do that, as soon as the data leaves your, a computer that you own and you put it on a server, it's subject to the whims of whoever owns that server, right? And you don't, sometimes you trust them, but it's hard to say whether you can. And if that can be problematic if you've got sensitive data of some sort, like patient data, for example. So is there another way of setting up a web application that avoids all of these problems? Now, I wouldn't be standing here if I didn't have an answer for you. And the answer is yes, there is another way to do it. And that is to do all the work on the client machine without ever involving a backend server. So this is possible because the browser itself actually provides a programming environment. So despite all the fun that we make of it, JavaScript is an actual programming language that you can use in actual useful applications. And at least for me, once I got my head around the asynchronous programming concepts, it was pretty easy to use. The browser provides a whole bunch of standard APIs for caching and all this various things. And for everything else, there's a very large ecosystem of third-party packages, which is sometimes you hear people talking about NPM, and that's basically a repository for it's the package manager for JavaScript stuff. However, for our applications, the real game changer was the rise of WebAssembly. So what is WebAssembly? So WebAssembly, or Wasm for short, is, begin quote, a binary instruction format that can be portably executed by browsers with near native performance, end quote. Okay, but what does that actually mean? Well, it means you can take code that's written in C or C++ or some other supported languages like Rust and Go, and you can compile that code into a WebAssembly binary that can then be executed inside the browser. And the near native part means that the code that is running in Wasm in the browser is almost as fast as if you had taken the same C or C++ code and compiled it to a native executable that you ran on the command line, for example. So that's really cool because it means that we get this high performance code without requiring the user to install anything on their computer, because WebAssembly just runs directly in the browser, which is already installed. Another very nice thing is that WebAssembly itself is also naturally interoperable with JavaScript code. So you can just basically plug it into existing web development frameworks with just a little bit of effort, and it all kind of just works. So now that we have this WebAssembly technology, we can take scientific libraries for doing data analysis that were written in C or C++, compile them to WebAssembly, and then perform the entire analysis inside the browser. This idea is inspired by the BioWasm project, which does this exact treatment for HTSLib. So for those of you who don't know, HTSLib is the C library that underlies SAM tools. So by doing so, basically the BioWasm people were able to operate on BAM files and manipulate BAM files inside the browser. So the general idea here is that if we can get this, if we can compile these libraries into Wasm, the browser can manage the entire analysis without ever having to transfer data to a backend to do the calculations. And if we don't need a backend, this allows us to eliminate the whole class of problems related to cost and latency and data ownership and so on. That brings us to the topic of the talk today, which is Kana. So Kana is a web application that Jay and I have been developing for analyzing single-cell data in the browser. And the long story short here is that we took some C++ libraries for single-cell data analyses, compiled them to WebAssembly, and then wrapped this all in a nice user interface to provide this point-and-click analysis experience. So we support basic analyses of single samples, merged analyses of with batch correction of multiple samples, as well as site-seq analyses involving protein and RNA readouts. So you can see the app as it runs on the left here. I'm just going to wait until it cycles around. One of the things about GIFs is that you can't really control when it triggers, but in a few seconds, I'll eventually decide to restart the analysis. Okay. Once I finish playing around with it. There we go. Okay. All right. So, basically a user picks a file that's on their laptop or desktop or whatever, and then they add the file, and then they press analyze, and then you can see the results start showing up straight away because there's no need to transfer the data to any backend server. Everything is computed on the fly inside the browser. I'll talk a bit more about this user interface in the next few slides, but if you don't want to hear me drone on for the next 30 minutes, you can actually just try it out yourself. At www.jcache.com/kana. So if you want to try it out now, you can. It's best to do this on a laptop or a desktop though, because the user experience is definitely not optimized for mobile. But if you're feeling adventurous, you can actually get it to run on your mobile device. So here I am running a single cell analysis inside my mobile phone. It took about 2 minutes to analyze 3,000 cells. So I mean, it's just a free mobile from that I got with my phone plan. But yeah, it turns out that you can use mobiles for more things than playing games. So you can actually use them to analyze single cell data as well. Right. so kana provides this sort of one-click analysis experience for single-cell data. The sort of terms and conditions there says give or take a few clicks to actually select the file that you're interested in. But you get the idea. You don't have to click that much, and all the results start showing up. And it just computes all the usual results that you expect from a standard analysis. So we have the marker table on the right. Yeah, on the right, a marker table on the right. We have the main panel with our beautiful unicorn vomit on the top here, and then we have this gallery section on the bottom that contains various bits and pieces of other plots that we compute as part of the analysis. So this is a pretty standard layout for a lot of single cell applications. Everyone expects to see these results, and there's not that much space on the screen, so you can't be too adventurous with your layouts. So as a result, every single cell application kind of looks the same, but that's okay. As for the main panel with our reduced dimensions, we have all the standard features. So you can zoom and pan, you can make lasso selections. I'll talk a bit more about that in a moment. Another feature that we stole from an application called Cirrocumulus is the ability to save to a gallery. So you can save different views of your reduced dimensions that are colored by different things, like in this case colored by different marker genes, but you can also color by different covariates. So you can save them into this gallery section, so you can look at them simultaneously if you have a big enough screen. I have a curved monitor at home, so I do have a big enough screen. We also have the least requested feature, which is the ability to animate your t-SNEs and UMAPs. So what we do is we catch the coordinates of the t-SNEs and UMAPs at every iteration of the analysis. And then you can get these sort of nice smooth GIFs that you can show as a party trick. Yeah, it's not really, I don't know how much scientific value it has, but we could do it, and we did. It's best to do this on a fairly small data set. Otherwise, it looks like you're like watching YouTube with dial-up. But if the data set is small enough, it looks pretty smooth. Okay. Continuing on with these reduced dimensions, another thing that we added was the ability to do this automatic cluster highlighting. So oftentimes, like in this case, it's not too bad, but in some cases you have these big t-SNEs and UMAPs with like 50 different clusters and 50 different colors, and it's kind of hard for the human eye to determine which color is which after a while. So what we do to make life a little bit easier is that whenever you mouse over a particular part of the plot, the corresponding label on the legend will also be highlighted. So it's a bit easier to figure out where you are and which cluster you're looking at. And just in case you're not sure, you can actually just double click on a particular cluster and it'll gray out everything else just like that. So it's just a sort of quality of life thing that we added to make it easier to look through very complex plots. Another cool thing that we added is the ability to dynamically adjust the color gradient to highlight areas of contrast in the plot. So in this example, we have the marker genes for cluster 7, and you can see the top ranked marker is this ribosomal protein. There's a gene I mentioned, ribosomal protein gene. So as you might expect from a ribosomal protein, it's basically expressed everywhere. So when you click on it, it's just all blue, right? And it kind of looks crap. And it doesn't look very convincing as a marker gene. But what you can do, I keep forgetting where to turn. What you can do is you can actually adjust the color scale so as to highlight the differences at the high expression range. So here I'm adjusting the color, and you can see that indeed there are actually differences in expression for this gene across my different clusters. So keep in mind that this adjustment here, so this color scale is in log 2 units of expression. So even after adjustment, the white is at, okay, when it gets back to it, the white is at 3. Wait, let's see if I can catch myself doing it. Yeah, the white is at 3 and the blue is at 6. So the difference between white and blue here is still an 8-fold change in expression that we would otherwise kind of not see if we just left the color bar as it is. So I find this feature quite useful when the markers are a bit more complicated than expressed and not expressed, for example. The other major part of the application is this marker table, and there's not too many surprises here. Yeah, this isn't a GIF. There's not too many surprises here. So this is you can pick the modality. In this case, we have RNA, but if you have site-seq data, you can switch between the RNA and AEDT markers. You have the cluster of interest, and you can rank the marker genes by different rank statistics. So we actually have a whole bunch of different ways to rank markers because we use the scran R packages methods for finding markers for each cluster. So how scran does it and how we do it in kana is that we for each cluster of interest, we compute we do a pairwise comparison with every other cluster. And then we compute a bunch of effect sizes from that pairwise comparison. So here are a couple of the effect sizes that we support. Then for each effect size, we aggregate the effect sizes across all the pairwise comparisons involving that cluster into a single ranking. And depending on which effect size you choose and which aggregation strategy you choose, you can get different sets of markers. So for example, if you choose the delta detected effect size is basically the difference in the proportion of cells that are detected with expression across different clusters, right? So if you want to rank marker genes by the delta detected, then you favor markers that are basically expressed in your cluster of interest and completely silent in the other cluster, for example. And the choice of aggregation also affects the markers. So if you use the min rank aggregation strategy, then basically you will favor markers, you will select for markers that are expressed in your cluster of interest compared to any of the other clusters. Whereas if you select the minimum aggregation strategy, you will select for markers that are expressed in your cluster of interest compared to every other cluster. So it's like an any versus all scheme, a policy that you use for selecting markers. And the mean is somewhere in the middle. But basically, there's a bunch of different choices that you can use to rank markers here. And if you want to do something more along the lines of filtering, you can also just filter directly, either based on the log-fold change. You can set a threshold there, or you can say, hey, I don't want any genes that are more highly abundant than some certain other threshold. You can set that here, and that'll automatically filter the set of genes that are shown. Yeah, I did mention lasso selections before, and here it is in action. So you can click the selection tab at the top there, and then you can select a thingy, and then you can save this. GIF's going too fast for me. Okay, yeah. Well, all right. Well, I'm just going to wait till it cycles around again. Okay, all right. Okay, here we go. Okay, select, and then you draw a lasso here, and then then you can press the save button to save that particular selection. Then I'm just clicking on it. Yes, indeed, I saved that selection. And that will automatically compute markers for that selection against all other cells in the data set. And you can see that in this case, we have this particular selection is upregulated for some HLA genes, which sound pretty important. So it's good that we found it. Yeah, okay. The final sort of marker-related thing I'll talk about is this versus mode for comparing specific pairs of clusters. So this is useful mostly when you have neighboring clusters in a plot or closely related clusters, and you want to figure out, hey, why did the clustering algorithm decide to split my big blob into a whole bunch of different clusters? What's the rationale here? So you can use this versus mode to say, hey, what are the difference between these two clusters that I'm interested in? What causes them to be separated? So in this particular example, I'm comparing these two clusters, so cluster 4 and cluster 2 here. And when I go into this versus mode, I can basically see the comparison between these two clusters directly. Let me see if it cycles back. Okay, so cluster 4 is this dark green one, and cluster 2 is this dark blue one. So here I'm picking cluster 4, and then I select versus mode. Then I go to cluster 2, and that gives me the markers for cluster 4 versus cluster 2. And then if I click that button in the middle, then I switch the order. So this is the markers for cluster 2 versus cluster 4. And then you can see in this case, the markers were in the AEDT modality. So yeah, it's whoops. Yeah, so that sort of makes it easier if you want to do some more in-depth investigations of why the clusters are being partitioned as they are. All right. Yeah, so the final few things I'll talk about is, so yeah, as I mentioned before, single cell data analyses are often quite iterative, and kana tries to optimize this analysis loop by basically caching the results of a previous analysis. So for example, if a user wants to rerun the analysis with more PCs, kana will detect that, hey, in this new round of analyses, the only thing that has changed is the number of PCs that the user is requesting. So it's smart enough to avoid recomputing any of the previous steps and just pick up from the cached results from those previous steps in order to perform the PCA and perform all of its downstream steps. So in this manner, we can more quickly, or the user can more quickly iterate across different analysis parameter combinations, because basically at every cycle, we're just reusing some cached results. And this is particularly nice if, for most people, what they'll be changing is the clustering resolution most of the time. And in that case, the only thing that kana needs to recompute is the clustering, because everything else is cached. You can also save the analysis state for later. So there's an export button up the top, which allows you to either save the analysis state to the browser's cache, sorry. And then that allows you to reload the same analysis in a different tab or in a different browser session. You can also download the analysis state to file. So you can archive that, or you can send it to a collaborator, and then they can open up the same analysis that you were looking at. Here's a sort of example of one I prepared earlier. So if you go to load saved analysis, you can actually pick a file on your computer. Here's one I prepared earlier, this kana file. And then if you press analyze, it doesn't actually analyze things. It just loads all the data and all the results from file. So you can see how quick it was, right? So it just actually has to read them from file and then just recreate the analysis state. So yeah, that's pretty nice for being able to share your analyses with different people. Okay, the final thing I'll mention is how extensible kana is for different inputs. So we support the major file formats, so matrix market, HDF5 files produced by the 10x genomics cell ranger pipeline. We support H5AD files that are produced by from the Python world. We also support RDS files containing summarized experiment or single cell experiment objects. So this is actually not quite as easy as it sounds, because remember, we don't actually have R running anywhere. So getting this to work required actually writing an RDS file parser so that it works in the browser. So that was I'm pretty proud of that little bit of reverse engineering, but that was pretty wild. And also reads for some data sets, we can also read them straight out of Experiment Hub. So Bioconductor's Experiment Hub, we actually interface with their REST API, and we can pull down data sets. So if you don't have if you want to try it out, but you don't actually have a data set on your computer, you can just load one from Experiment Hub. And that's that works for some curated data sets from some classic papers. And it kind of allows you to mix and match between different formats. So you can load one data set in matrix market, and you can load another one in HDF5, and kana will automatically know how to take the intersection and do batch correction and combine both data sets into the downstream analysis. All right, so I think that's all the features I was going to talk about. So now now that you know what it is, I'm going to tell you about how we did it. So if you recall, WebAssembly requires us to have C++ code or some other supported language, but our analysis pipeline for Bioconductor is written in R. So what we have to do is we have to do this sort of airlift of R code, of the R analysis code, out of R and into C++. So this is not as difficult as it sounds, even though it does sound fairly difficult, but it's not as difficult as it sounds, because at least for the Bioconductor single cell workflows, a lot of the heavy compute is actually written in C++ anyway. So in that case, it's just a matter of taking out the C++ code from inside the R packages and just refactoring them so that they work as standalone C++ libraries. So yeah, it's not that bad, but it still is a lot of work. And this list here represents several weekends, or a year's worth of weekends of working in order to get all the packages, get all the libraries that we need into C++. So each entry on this manifest here is one library that I had to create for this project. And I'm not going to go through the entire thing, but I will mention a couple of highlights. So the IRLBA library for doing the PCA, that was pretty wild, because I didn't really understand any of the math when I was translating from the R package. So it was kind of like if you have to drive a car, but you've taped over all the windows, so you can't actually see where you're going. It's theoretically possible, but it's a really wild experience. So I'm very by trial and error, I eventually managed to get it to give the same results as the R package. So that was wild. The k-means was also pretty wild, because that involved a translation from the R's Fortran implementation. So that was like, wow, that was like blast from the 80s. So that was also pretty crazy. Yeah, the final thing I'll mention is Tatami. I'll talk a bit more about this in a few slides, but Tatami is the spiritual successor to the Beachmat R package that provides access to various matrix representations. And the sort of extra part that it builds on beyond Beachmat is its ability to support delayed operations in C++. So yeah, I'll go through this more in a few slides. Okay, anyway, once we have our C++ libraries, we can start to think about making a WebAssembly binary. And how we do it is we use the Emscripten toolchain, which takes C++ code and compiles it into a Wasm binary. So it does this using Clang and LLVM under the hood. And all of this is orchestrated using CMake. So at the end of the day, we get a WebAssembly binary that is 3 to 4 megabytes in size. And that's actually, to me, that's really impressive that we can fit the entire single cell analysis stack, all the way from loading count data, all the way to reduced dimensions within a 3 to 4 megabyte software package. Right? For comparison, if you have a look at the Oscar Docker image, so the Docker image that's used to build the Oscar book, that's at least a gigabyte, right? So getting that similar amount of functionality in 3 to 4 megabytes is pretty impressive. Yeah, anyway, once we have this Wasm binary, we then wrap it inside a JavaScript package to provide a nicer developer interface. And this JavaScript package is called Scran.js, because what else could it be called? And that's what's published on NPM. So if you want to, if you ever feel the need to do single cell analyses in JavaScript, you can actually pull this package down and start working away. Yeah, but once it's in NPM, it's a very straightforward process to incorporate it as part of the standard React build process. So for those of you who don't know, React is a very popular framework for developing single page applications. Yeah, we just use the same sort of setup. So we pull down Scran.js and incorporate it into our web app. And I'm not going to talk too much of it, or at all actually, about the web app detail, about the sort of user interface details, because that's more of Jay's wheelhouse. But yeah, at the end of the day, we end up with Kana, which is the interface that I showed you earlier. This is a provisional logo. We're still looking for a better logo. So if anyone has any ideas, we're happy to take a PR, right? Yeah, but that's basically how it's done. And I think it's actually fairly, it more or less just works. I think the trickiest part was managing the data transfer or the data sharing between JavaScript and WebAssembly. But once we figured that out, the rest of it was actually pretty straightforward. So yeah, it was pretty much, in terms of developer friendliness, it was pretty much as advertised. All right, okay, but that's enough talk. What does the code look like? So on the left here, we have the bioconductor, the sequence of R function calls for a bioconductor-based single cell analysis. So if you read the function names, they pretty much just say whatever. It explains itself. On the right here, we have the corresponding sequence of corresponding code for the analysis using Scran.js in JavaScript. And you can see that it's basically mirrored from R into JavaScript. So you have the same type of functions like compute QC metrics, log norm count. In fact, the names of the functions are pretty much the same because I don't really have much of an imagination. So I just used exactly the same function names. So hopefully you can see it's actually a fairly faithful translation of the original R workflow into JavaScript that we use in our that we ultimately use in the web application. However, there are a few tweaks that we have to do to get it to run performantly. Is that a word? To run at high performance in the browser. And the first of these tweaks is motivated by the fact that WebAssembly, at least most implementations of the WebAssembly specification, uses 32-bit pointers. And that means that with 32-bit pointers, you have 4 gigabytes of addressable memory space in the WebAssembly heap. So in simpler terms, it means that a program that you compile to WebAssembly in 32-bit mode has 4 gigabytes of memory that it has access to, right? And that's not a lot. right, for squeezing our big single cell data sets into this little 4 GB space. So we have to do some tricks. And the main trick that we use here is this concept of a layered sparse matrix, where we split our matrix, our count matrix by row into different integer storage types. So the idea here is that for most low abundance genes, their maximum count for across all cells for that gene is going to be less than 256, right? So if it's a gene with a maximum count that's less than 256, we can store its count as an 8-bit integer. Similarly, if it's greater than that, but the maximum count is still less than 65,000-ish, then we can store it as a 16-bit integer. And anything bigger than that, we have to store it as a 32-bit integer. So with this strategy, we can store most of the count matrix in 8-bit integers and save ourselves basically a 4-fold reduction in space compared to storing everything as a 32-bit integer. So that's one way we compact down the data set. Another way we do it is using these delayed operations that I talked about earlier. So the analysis involves several whole matrix operations, like basically operations that would naively result in the creation of another matrix. So QC filtering, you have to subset the matrix and create a new matrix that has the subset of cells that you want to keep. Normalization, you divide by the size factors, you create a new matrix with the counts divided by the size factors, and then log transformation, you create a new matrix. So if every time we actually created a new matrix, we'd quickly run out of space. So instead, what we do is we use for each of these sort of whole matrix operations, instead of creating a new matrix, we actually wrap our existing matrix in a delayed operation that represents that particular operation. So we have a delayed operation that represents the QC filtering. Then we wrap a delayed operation that represents the normalization, and then we wrap another delayed operation that represents the log transformation. So we never actually make copies of the matrix. There's only actually ever one copy of the original data set, and each of these analysis steps, we basically mimic the creation of a new matrix by wrapping it in a delayed operation. So that's pretty much what the delayed array package does from in the bioconductor world, and we basically brought that across into our WebAssembly implementation so that we can save a bit of space. And of course, when we do the PCA, instead of sort of naively centering and breaking sparsity and converting a sparse matrix into a dense matrix, which would be unacceptable, we defer this centering to the matrix multiplication step so that we never actually realize a dense matrix in memory. So that's a fairly standard trick, but we also do that as well. In addition to that, another tweak that we have to do is to deal with parallelization. And that's because parallelizing in the browser is a little bit weird. There's no OpenMP or stuff you can rely on there. Instead, there's this thing called web workers, which are basically JavaScript-based threads that are managed by the browser, right? And Emscripten is really cool because it actually naturally supports parallelization by these web workers. So as long as you configure your C++ libraries to use the standard threading library instead of OpenMP, Emscripten will automatically convert your POSIX threading calls into web worker messages. So it just does this for you automatically. You don't actually have to do it yourself. And that's really nice because it allows kana to use multiple CPUs on the client machine for doing various things. So for example, if we have a count matrix that we want to process, each worker works on a different chunk of the matrix, and then we can get everything done quickly. So we do this for the sparse matrix multiplication. So we chunk across rows or columns depending on which side we're multiplying from. We do this for nearest neighbors searches. So each worker searches for the nearest neighbors of a particular chunk of cells. And we do this for marker detection. So each worker works on a different set of genes. So this is a nice way of parallelizing that gets us a more performance for the client. Of course, some steps aren't easily parallelized, like the clustering implementation that we use from iGraph. At least I can't figure out if it's parallelizable. TC and UMaps also are difficult to parallelize because each iteration depends on the previous iteration. So it's not like you can really split them up. So in those cases, instead of trying to parallelize within each task, we parallelize across tasks because none of these things depend on each other. So we can just run all of them concurrently, get greater CPU utilization, and reduce the runtime of our analysis. So with all these performance tweaks, how does Wasm actually do in terms of performance? Is it near native as claimed? Well, the answer is sometimes, which is probably the least satisfying answer, but whatever. So to test this, what we did was we took kana and we timed how long it took for kana to run through the analysis, starting from count loading all the way to reduced dimensions. And we do this for a bunch of different data sets. Then we took the same C++ code that we used to create the WebAssembly binary. We take that C++ code and then we compile it into a native executable that we can run on the command line. Then we just repeat the timings for the native executable, and that's the timings you see on the right here. So if you compare Kana's timings with the native timings, you'll see that Kana is about 25 to 50% slower. I'm not sure how much of this is due to WebAssembly and how much of this is due to sort of other JavaScript-related inefficiencies. It's not quite near native, in my opinion, because near native is more like 5 to 10% slower. But yeah, it doesn't really matter. It doesn't really matter because if you look at the times here, these times are in seconds. So for 100,000 cells, this takes about, what is it, 4 minutes? 4 minutes to analyze in the browser. So I think that's plenty fast, right? That's like, you can go get a coffee and come back and it's fine. Reassuringly, the memory usage stays below 4 gigabytes here. So that's also good. So we don't blow through the Wasm heap limits. So hopefully I've been able to convince you that it is possible to efficiently analyze single cell data inside the browser on this sort of client-side compute paradigm. But just to recap, why are we doing this again? Right. So the most obvious reason is that we don't have to have a backend, as I mentioned before. We don't have to worry about cost or paying for someone to watch the server or having uptime guarantees or DevOps stuff. We don't have to deal with any of that, right? We don't have to transfer data, so there's no latency, there's no bandwidth issues, there's no worries about ownership. And one of the things that's kind of underestimated is the fact that this client-side compute strategy actually scales to any number of concurrent users. So if we had 1 million people using Kana right now, we could probably handle it. If GitHub pages can handle 1 million accesses at once, then we can probably handle it because each user brings their own compute to the party, right? The application maintainer is not responsible for provisioning, server capacity or EC2 instances in order to get the analysis done. So this is quite nice, at least especially from an application owner's point of view, because it means that I don't get any sort of bill shock from cloud auto scaling when a whole bunch of people decide to use the application after they hear some kind of presentation about it, for example. Of course, it's not a free lunch. There are its own, like this strategy does have its own challenges, and the most obvious of these is that you're subject to the resource constraints on the client machine. So this is hard limitations like the number of CPUs that are available, the amount of RAM that's available, but there's also soft limits. So for example, Kana will only ever use 2/3 of the CPUs that are available on the user's machine. And that's to keep some CPUs free for the user to do other things while the analysis is running. So I like to watch YouTube, and it's hard to watch YouTube and analyze data if the data analysis is using up all the cores. If your data lives on a remote database, then you need to transfer files from the remote database to the client in order to do the compute. And then in that case, you get all these issues with bandwidth and ownership, resurfacing again. And the final thing is that if anyone's ever developed for a web page, the browser is a very strange place to do program software development. And there's a lot of security restrictions to avoid web pages doing bad things to you when you open them. So doing what you want while keeping the browser happy usually requires a little bit of ingenuity. But yeah, that's kind of something that you have to deal with. So yeah, that's kind of how it is. But there is a way that we can get the best of both worlds. And this stems from the fact that we originally wrote all of our code in C++. So we can basically take the same C++ libraries and then compile them to some compilation target that can run on the server. So for example, the same C++ libraries that are used in Kana via WebAssembly can actually be used by R packages. So you can take that same code, wrap it in RCPP, and deploy it as an R package. And that's exactly what the Scranchan package does. So it gives you the same analysis that Kana provides. And in fact, the single R bioconductor package for cell type annotation has also switched to the pure C++ implementation under the hood. So it shares that implementation with Kana. More simply, you can actually take JavaScript and run it on the back end in the server using Node.js. So Scran.js will happily run in a node context. And this means that you can use Scran.js to set up a sort of backend service that does the computation and sends results to the client. And in fact, that's exactly what we did. So Canopy provides a more traditional backend for Kana, where Kana is more like a web page front end, and it can send a request for Canopy to do the compute. And yeah, if you don't want to deal with any of these damn newfangled languages with garbage collection, you can actually try using the command line tool that we used for timing. So that also is another option if you want to try it out. So anyway, the moral of the story here is that if you write your analysis code in a low-level language, you can have this sort of philosophy of write once and deploy everywhere, where you write it once and then you can compile it to different targets that can run basically anywhere else. And this leads to the the idea of hybrid compute, where the Kana application dynamically chooses its compute location based on the nature of the data. So for example, if your files are small, or if the file already lives on the client machine, or if they're coming from a remote database, but they're very small and they're easy to distribute, or you're allowed to distribute them, then you can do an in-browser compute, so a client-side compute, just like Kana currently does, in order to take advantage of the sort of high per-user scaling. On the other hand, if the data is large, it's like very difficult to transfer, or you're legally not allowed to transfer it out of a particular jurisdiction, then it makes more sense to have everything running in a backend where you can have greater control. So either running on-prem or using AWS or whatever. So yeah, this provides a nice way of sort of having our lunch and eating it too, right? In terms of getting the best of both the sort of client side and the backend strategy. And we're starting to explore this for some of our in-house databases that manage our internal data. All right, so yeah, that's pretty much it. I'll just mention a couple of the things we're thinking about doing. So obviously, for any web application, we spend so much time doing this UI, UX stuff. So we'll probably be spending a lot of time doing that again. We're thinking about doing some web GPU stuff to use the GPU to do some faster compute. I'm not 100% convinced on that, but it's an option. More modalities, more input sources. We recently open sourced this ArtifactDB thing that people here might be interested in actually. So we can also pull data from there into Kana. Better out-of-browser tooling. So I mentioned before that you can actually dump the analysis state. So we actually have a Kana parser package that will open that analysis in R. So basically that you can have a collaborator who does the analysis, and then you can send it over to an analyst who actually opens it in R to do other things, right? So that's a pretty nice way to interoperate between different worlds. Yeah, and I guess we should submit the manuscript somewhere, but it's on bioRxiv. So if you go to bioRxiv and look up Kana, you'll probably find it. Yeah, it's not a major priority. But anyway, so just to wrap up the acknowledgements. So this is a joint project between me and Jay. So these are us two bros on the mountaintop in sort of north of the city. Yeah, also thanks to everyone in the team, especially Mike and Hector for looking the other way while we worked on this. So yeah, and then thanks to our power users, which is currently mostly Pete, for giving us feedback on some useful features. And yeah, so yeah, that's pretty much it. Hopefully I ran on time, I'm not sure.
Sorry, clicks, am I on? Sweet. Questions in the room? Oh, I regret this already.
Thanks for the great talk. What recommendations do you have for developers for capturing the reproducibility aspects of the interactive analysis that happens, and for reporting purpose or for future provenance of the analysis?
Right. So at least in Kana, what we do is all of the analysis state is tracked. Basically, there's only one place that people can enter analysis parameters, right? And we basically take a snapshot of it every time someone clicks the analyze button. So that particular analysis state is saved. And that's in terms of the parameters. The results are a bit tricky to guarantee reproducibility, because I've often tried this myself, and it's actually a lot harder than you might think because different machines have different numerical precision, and that's very hard to guarantee. The best that we can usually do is to make sure we record like the environment in which it ran, the parameters, the environment where it ran, and just keep, hold on to the results somewhere. And that's usually been good enough. People are fairly understanding if they don't get exactly the same result. The t-SNEs and the UMAPs are pretty wild because small deviations can give you completely different results. So that's a bit harder to guarantee reproducibility. That's what we do. Yeah.
One from online from Dario is, how is it being used within Genentech and Roche?
So we use it. We use it mostly in two modes. So inside Genentech, we have our own sort of internal mode, which is a light mode that does that just views the results and doesn't do any analysis. And then we have the full-fledged mode that actually does the analysis. So those are we don't have the sort of light mode made public yet, but that's mostly how we use it. And the only major difference is that the Genentech version pulls from internal databases. So a bit of what I was mentioning earlier. We pull from our internal databases and results that contain single cell data. So it's not just limited to public sources or the sources that are on the client machine.
Thanks, Aaron. Also, nice to see you again.
Pleasure to see you as well.
Nice. My question, I think you mentioned a little bit at the end about the sharing. For example, if you wanted to share this analysis with someone. So forgive my ignorance about how this works. But if I'm doing an analysis on my browser, can someone else share it, or do I just need to do them the R files and?
Right. Yeah. So in the very simplest mode, it would be something like, hey, I want to save my analysis state. I have the save files, and then I just Dropbox or email or whatever, transfer it to someone else to use it, right? Inside the company, we have a more sophisticated way because we have our own infrastructure for file sharing. So that export button, if I remember correctly, that export button has an extra button that says export to like results DB or something like that. And then on the other side, whoever else can then just pull it down from results DB when they want to load a saved analysis. Doing that for public, for people in general, for just public usage is a bit harder because then you have to figure out how to pay for that file store. But at least at the organizational level, it's definitely possible to extend kana for a fairly, if you have a large organization with people who want to use kana to share information, then it's definitely possible to extend kana to support custom data and analysis transfer schemes, right?